This is part 3 of the Chapter 7 lecture notes, and we left off with slide 19. I didn't get very far with it, so just to kind of review what we've covered so far, photosynthesis has two stages or two parts, and the first part occurs in the chloroplast in the thylakoids, and they are called the light reactions. So when you're learning the light reactions, you're really learning kind of how a machine works. You know, it's more like process. The processes that occur in order to generate ATP and NADPH, because that's the whole point of the light reactions, is to produce the ATP and the NADPH that are going to fuel or power the Calvin cycle reactions. But the light reactions are more of a mechanical type process that you have to learn how it works. And the Calvin cycle reactions are just a series of chemical reactions. So I really focus, when I teach the light reactions, I focus on explaining to you how the process works, you know. So looking at the thylakoid membrane, so here's the thylakoid membrane. You can see where it was cut away here. So it's just like the edge of one of the thylakoids, and you can see inside the thylakoid space, and outside of the membrane, out here in this open space, would be the stroma of the chloroplast. So during the light reactions, the first thing that's going to happen is photosystem 2, which is this big orange protein molecule right here, that also consists of many pigment molecules. But photosystem 2 is going to absorb or capture sunlight energy. And that energy is going to excite an electron. And this electron originally comes from water. But anyway, that electron, once it is excited by sunlight energy, is going to first reach a reaction center chlorophyll A molecule, which is not shown here, but just we went over it before. Then it's going to arrive at an electron acceptor molecule, and that's when it will leave photosystem 2. It will leave photosystem 2 with very, very high energy, and it will pass through these, what we call electron carriers. And you can see them here. Here's one, and then here's another one, here's another one, another. And finally, it is used to produce NADPH. After the electron reaches photosystem 1, and sunlight is absorbed and excites that electron again, then the energy from that electron is going to be used. It's going to combine with hydrogen ions and NADP plus to produce NADPH, which will go on to the Calvin cycle reaction and help fuel that reaction. So what we just saw happen, by the way, is NADP plus gained electrons. And remember back to oil rig. It will let me go backwards. Hold on. Where did I write oil rig? Here it is. Okay, I made a line through it, but you can still see it says oil rig. Oxidation is loss, reduction is gain. So what's happening is that the NADP plus molecule gains electrons along with hydrogen ions, so that means it is reduced. So what we're seeing happen here is a reduction reaction. Now, the other thing that happens is the energy from those electrons flowing from carrier molecule to carrier molecule down that electron transport chain. And this is the part that I keep going back and forth. This is the electron transport chain part, the part between photosystem 2 and photosystem 1. So the flow of electrons is releasing energy. That energy is used to pump hydrogen ions into the thylakoid. And all these hydrogen ions build up in here. Well, we've learned what happens due to a concentration gradient. when you get a very, very high concentration of anything on one side of a membrane, then if there's an opening or a point where they can travel, they're going to move out. They're going to move from where their concentration is high to where it is low. And that's what the hydrogen ions do. They build up. So the flow of electrons generated the energy to pump hydrogen into the thylakoid. Once hydrogen builds up enough of a concentration gradient, these ions will eventually encounter the ATP synthase molecule. And they're going to want to flow out because they're going to move down their concentration gradient from high to low. So that flow of hydrogen ions is the energy that's used to generate ATP. And that is what chemiosmosis is. Chemiosmosis is when you're using a concentration gradient, when you're generating a concentration gradient and using that concentration gradient to create a molecule or to catalyze a reaction. So the important components of the electron transport chain are the flow of electrons, the pumping of hydrogen across a membrane so that it builds up a concentration gradient, and then the flow of hydrogen ions and that energy being used to generate ATP. We're going to see this again in Chapter 8. The electron transport chain works the same way, whether or not it's a part of photosynthesis or cellular respiration. But anyway, we can see that here's our two photosystems, photosystem 2, photosystem 1. We can see our important molecules that are produced are ATP and NADPH. And so now we're going to talk about ATP production in the notes, highlighted in yellow. The transfer or flow of electrons down the electron transport chain yields energy. The energy from the flow of electrons is used to pump hydrogen across the thylakoid membrane into the space. Then once hydrogen encounters ATP synthase, it has a channel that it can flow through. So it's going to flow back across the thylakoid membrane to where its concentration is lower. And that ATP synthase is going to produce ATP using the energy from the flow of the hydrogen ions. This method of producing ATP is called chemiosmosis. Now, what comes next, and it's not, the slideshow is not real clear. In section 7.4, what we're really doing is we are covering the Calvin cycle. So I'm going to take a chance and try to write it. So this is where we start. We just covered the light reactions. They occur in the thylakoids. So now we're going to cover the Calvin cycle reactions. Let's see, goodness. C-A-L-V-I-N. Calvin cycle reactions. So the Calvin cycle reactions are the part that convert carbon dioxide into food or carbohydrate. There are 3 stages to the Calvin cycle reactions, and there's a lot of chemistry in this, and I try to simplify it as much as possible, just so that you can get a good understanding of an overview of what's happening. I don't necessarily expect you to understand all the chemistry or to even memorize all the chemistry, but I want you to know basically what's happening. The big picture. So carbon dioxide fixation is step 1, carbon dioxide reduction is step 2, and then RUBP regeneration is step 3. So let's start with step 1 of the Calvin cycle, carbon dioxide fixation. The most important thing to know is that carbon dioxide is fixed or attached to a molecule called RUBP. Trying to make sure I say it correctly, ribulose biphosphate, I believe is how you say it. But we're going to call it RUBP. The enzyme that catalyzes this reaction is RUBP carboxylase, or it's more commonly known as Rubisco. Now, so you will understand how important this enzyme is, this enzyme Rubisco is the most abundant enzyme of all the enzymes that there are. It is by far way above and beyond more abundant than any other enzyme that exists. And so that just lets you know how important photosynthesis is. So this enzyme RUBP carboxylase catalyzes the reaction where carbon dioxide is attached to RUBP, attached or fixed to RUBP. Now, once that happens, and now it says Calvin cycle. Once that happens, it's then got to be converted into a molecule called G3P, which is the molecule that can be used to produce glucose. So as you see here, the Calvin cycle reactions occur in the stroma. They take carbon dioxide from the atmosphere and convert it into a carbohydrate. And what we're seeing happen in this slide is that the carbon dioxide molecules are being fixed or attached to RUBP, ribulose 1,5-biphosphate. Now, the next few steps that you're seeing are the step 2, which is carbon dioxide reduction. And that step uses ATP and NADPH. I really don't care if you know every single chemical that's on this diagram, but I want you to know RUBP is the molecule that carbon dioxide is attached to. I want you to know Rubisco is the enzyme that catalyzes that reaction. And I want you to know the final product of step 2 is G3P, which is a 3-carbon molecule that can be used to produce glucose. And after that, then we have to rebuild, or we have to build back up our supply of RUBP. So that step is called RUBP regeneration. Okay, so step 2, the reduction of carbon dioxide. And that is when the molecule that is formed when carbon dioxide attaches to RUBP, it eventually is converted to G3P. And G3P is the product of the Calvin cycle reactions. G3P can be used to produce glucose or other carbohydrates or even amino acids or even fatty acids. This is the stage, step 2, the reduction of carbon dioxide. This is the stage where NADPH and ATP from the light reactions are used. Okay? Now, step 3 is the regeneration of RUBP, because we've used up the RUBP that carbon dioxide is fixed or attached to. We have to make some more. So we replace it by, for every time you make 6 G3Ps, I'll say it like this. This is the simplest way, I think. For every 6 G3Ps that are produced, 1 can be used to be food for the plant. The other 5 are used to regenerate RUBP. And the reason is because G3P has 3 carbons. So if you take 5 G3Ps times the 3 carbons, that's 15 carbons. 15 carbon atoms in 5 G3Ps. RUBP is a 5-carbon molecule. So if you take 3 RUBPs times the 5 carbons, that's 15. So that's why you have to have 5 G3Ps to regenerate 3 RUBPs, is because you want to make, you want to keep the number of carbons the same. The number of carbons in the molecules the same. So it takes 5 G3Ps to regenerate 3 RUBPs. So that's step 3, the regeneration of RUBP. Now, excuse me. The important